In this study, we looked at a specific type of kidney cancer called XP11 translocation renal cell carcinoma, or TRCC for short, which has been known for a while to have a female sex bias, and it's about twice as common in females as males. And it turns out that this type of kidney cancer, TRCC, is caused by a rearrangement of a gene called TFE3 that's located on the X chromosome. So we know that females have two X chromosomes and males only have one. And so that raised the question as to whether the reason why this kidney cancer was more common in females than males is because there are two opportunities to cause this rearrangement of the TFE3 gene on the X chromosome in females as compared to males. But there's actually a slight wrinkle in that the two female X's are not equal. So to maintain dosage compensation between males and females, one of the two copies of the X chromosome in females is epigenetically silenced. And that's a process that occurs early in development called X inactivation. So in this study, we performed whole genome sequencing on a cohort of TRCC tumors. And we found that when you look at rearrangements between the TFE3 gene on chromosome X and an autosome, those types of events are almost exactly twice as common in females as in males. And so basically this ascribes a genetic basis for the 2-to-1 female sex bias that we observe in this cancer. And we were further able to show that both the active X and the inactive X can be accessed for rearrangements in females. And when the latter happens, meaning a rearrangement between the inactive X and an autosome, we see evidence of long-range transcriptional changes, such as reactivation or reawakening of previously silenced portions of the inactive X, as well as potential silencing of portions of autosomes that become attached to the inactive X.
The paper highlights a conceptual gap in our understanding about cancer genes and cancer evolution because we haven't taken into account the difference between the inactive and active. I think this study really highlights the need and potentially therapeutic opportunities offered after we can separate those two types of alterations, because they're both named the chromosome X, but they're actually completely different. In fact, probably more different than you compare chromosome X to an autosome or other non-sex chromosomes. So putting them together is certainly a missed opportunity for understanding both cancer evolution and finding new therapeutic targets.